Trinity Cultural Academy, Coimbatore, and New India Publishing Agency, NEPUS, New Delhi, welcomes you all for this presentation on varietal seed production in maize tidbits 19, which comes under the major subject plant science and seed science and technology. And this material is highly useful for ICAR All India Entrance Examination, JRF and SRF for PG admission, and ICAR NET, ARS and STO, and also it is useful for IBPS AFO examination. And this material is prepared by Dr. K. Vanangamudi, formerly Dean Agriculture TNA Coimbatore and Dean Adiparashakti Agriculture College, Kalavai, and Professor and Head Seed Science and Technology, TNAU Coimbatore. And before entering into the seed production and maize aspects, let us see the floral biology of maize crop. It is a common millet in India with wider industrial and household utility, and it is used as a feed, food and raw materials in soft drink industries. Botanically, it is called as Zea maize and it comes under the family Poaceae, and it has a chromosome number 2n equal to 20. And the inflorescence is nothing but a panicle crop. As the crop is monoecious in nature, both male and female are present in the same plant but at different positions. There are two types of flowers, female and male. The female flower is known as carp, which is present in the axillary position and particularly in the middle of the plants. And the male flower is present in the terminal position, in a terminal inflorescence, which is known as tassel. And this one is a cob and this one is a tassel. And the next one is husk. It is an enlarged leaf sheath from each node forming a protective covering around the inflorescence. The pollination behavior here is cross-pollination, and particularly the androecium matures first, which favors the cross-pollination mechanism. Protandry is nothing but a androecium matures first, and the flowering pattern is top to bottom in case of tassel and bottom to top in case of cob, and the anthesis takes place in, that is, the pollen shedding begins 1 to 3 days before the silk emerges from the crop, and fertilization takes place within 12 to 18 hours after silk emergence. And the entire silk is receptive, which is pinkish or sticky at the beginning, but after fertilization it will become chocolate or brown in color. The number of pollen in each tassel is 2 crore 50 lakhs, and the pollen viability is about 12 to 18 hours only, but the silk is receptive for 8 to 10 days, and the male flower anthesis takes place in the time of 6 to 8 am, and the duration of flowering is 2 to 14 days. There are different types of maize, that is corn, as as per the classification of botanical characters. And the first one is flint corn, Zea mays indurata, dent corn, Zea mays indentata, and this is the most widely used corn. And sweet corn, Zea mays saccharata, Zea mays amylacea flour corn, and popcorn, Zea mays everata, and waxy corn, Zea mays serratina. The species characters are seen in breeding aspects, and there are different types of, in maize there are different types and methods of seed production, and there are open pollinated varieties, synthetics, composites and hybrids are available in case of maize crop. The first one is open pollinated varieties. Here the varieties are raised under isolation of 400 meter in case of foundation seed and 200 meter in case of certified seed, and here the plants are allowed to openly pollinate among themselves and seed set. And the next one is synthetics. In case of cross-pollinated species, a variety obtained by inmating all possible combinations. Here a number of lines is greater than 5 that will combine well with each other. The synthetic variety is baby corn BC1, that is baby corn variety. And the next one is composite varieties. These are produced by open pollination among a number of outstanding strains. usually not selected for combining ability with each other. Example here is K1, Jawahar, Vikram, Sona, Amber and Co1 and Kisan. These are the composite varieties. And the last one is inbreds. It is relatively true breeding strain resulting from a repeated selfing of 5 times. And the varietal seed production technique is nothing but, in case of maize, mostly the varietal seed production takes place under open pollination condition with the isolation is the common method of varietal seed production. There are different stages of seed multiplication. In case of maize varieties, composites and synthetics, it's multiplied by adopting 3-generation system of seed multiplication as breeder seed, foundation seed and certified seed. As the crop is highly cross-pollinated crop where the chances for contamination is very high, and the popular varieties of grain purpose are CO1, K1, CoH3 and CoH4, and for fodder maize African tall varieties, and for salad purpose CoBC1, CoBabycorn 1. And the season is the best season for production is June, July, November, December and January, February. And here the flowering should not coincide with either rain or high pH, and the maturation should coincide with the dry weather only. The temperature of 37 degrees Celsius is favorable for better seed setting, and the land requirement is the land required for open pollinated variety, composites and synthetics should be fertile, and the problem soils will lead to low pollen fertility and ultimately it affect the quality and the seed set will be poor. The previous crop should not be the same crop to avoid the occurrence of volunteer plants. If it is present, and it should be removed and it should be certified, has to be accepted for certification. So the field should not have any volunteer plants. The isolation distance for foundation and certified seed is 400 and 200 respectively. And the selection of seed. For production of foundation seed, breeder seed is used as a base material. For production of certified seed, foundation seed is used as a base material. And also the seed should be obtained from a authenticated source with a tag and a bill. The required seed rate will be 20 kg per hectare or 8 kg per acre. And the pre-sowing seed treatment. The seeds are given with any one of the seed rate is 20 kg per hectare or 8 kg per acre. And the pre-sowing seed treatment. The seeds are given with any one of the seed treatment or in combination. The seeds are soaked in 2% K2PO4 for 16 hours with a seed to solution ratio of 1:0.06 and dried back to the original moisture content of 8 to 9%. And this management could be used for both the dry land agriculture as well as garden land agriculture. And also it is treated with 5% carbofuran 3G to protect seed from shoot fly infection, and seed treatment with chlorpyrifos 4 ml per kg also useful for against the shoot fly. And the seeds dry dressed with bevistine 2 gram per kg of seed to protect the seed-borne pathogens and soil-borne pathogens. And seeds which treated with azospirillum 50 gram per kg of seed to fix atmospheric nitrogen. Any one of these treatment or combination of treatment is adapted for better productivity. Seeds also treated with a polycoat at 3 gram per kg of seed diluted in 5 ml of water, and it is act against for better marketability and production. Pink colored polycoat performed better than other color polymers. An adoption of sequence of treatment physiological should be followed with physical seed treatment. The sowing. The seeds are sown with a spacing of 45 by 10 or 60 by 20 at a depth of 2 to 4 cm based on the specific features of the variety. The nursery production will not be suited for this crop, and here in the main field the seeds are sown either in ridges or furrows or under beds and channels. The seeds are thinned and gap filled should be done 7 to 8 days after sowing. Here we can see the row to row spacing, and here we can see the plant to plant spacing. The spacing is given here 45 by 10 or 60 by 20. In case of varieties, the seed rate already we have seen the 20 kg per hectare or 8 kg per acre. The nutrient application at last ploughing apply 12.5 tons of compost per hectare, and the fertilization recommendation is 150 75 75 NPK kg per hectare, and it is applied in 3 split application, one as basal application 40 75 and 40, and the first top dressing is done after 20 days of sowing. It about 50 kg per hectare of nitrogen, and the second top dressing is on 40 days after sowing. There 60:0 is to 35 kg per hectare. And the micronutrient application here is 2% DAP is sprayed at 50% flowering stage to enhance the uniform flowering and increase the seed set. If zinc deficiency is found, apply 20 kg of zinc sulfate per hectare. If iron deficiency is found, apply 12.5 kg per hectare of micronutrient mixture. The crop is mostly affected by micronutrient deficiencies of N, P, and manganese and magnesium, zinc, iron and potassium. For this, 12.5 kg of micronutrient mixture is applied in furrows and mixtured in the soil. Weeding. Application of atrazine at 500 gram per hectare as pre-emergence herbicide, which controls the weed growth up to 20 to 25 days. If pulse crop is used as an intercrop, do not use atrazine. And one hand weeding on 17 to 80 days after sowing will keep the field free of weeds. In weeding after boot leaf stage is not economical, and shade will also minimize the weed flora. On organic production, 2 hand weeding at seedling stage and other at boot leaf formation, and other at boot leaf formation will keep the field weed free. The next one is irrigation. The crop should be irrigated once in 10 to 15 days for enhanced seed set and formation of boulder grinds. The critical stages of irrigation are primordial initiation stage, vegetative stage, flowering, milky and maturation stage. If the irrigation is withheld in these seed stages, seed set will be poor and seed size will be reduced. The next one is roguing. It is specific to seed crop and it is done from seedling stage to harvesting stage based on the phenotypic characters. The off types can be identified through stem color, plant structure, number of leaves, auricles, nodal color, tassel color, sheath color, grain color, etc. The field standards for a seed crop is as follows. The first important one in case of seed certification as a field standards are the number of inspection. The number of minimum of 2 inspections shall be made, one at flowering and another during flowering. And the isolation distance is about the maize field should be isolated from the contaminants, as the minimum isolation distance is for foundation seed 400 and for certified seed 200. And in maize hybrid alone, by increasing the border row, minimizing the isolation is permitted. Let us see there in maize hybrid seed production in clearly. And the specific standard is that the off type permitted for foundation seed is 1, and in case of certified seed is also 1. We all know that off type plants that have shed or shedding pollen at any one of the inspection during flowering, when 5% or more of plants and seed fields have receptive silk. Pre-harvest sanitation spray. Spraying of endosulfan at 0.07% or bevestin at 10 gram per liter 10 days prior to harvest prevent the seed weevil, that is Cytophilus varizae, infestation at storage. And the seed maturation takes place about at 14 to 20 days after anthesis. Milky stages occur, that is starchy fluid and stage, and 35 days after anthesis soft duff stage, 45 days after anthesis glazed duff stage, and after 55 days of anthesis ripe duff stage occurs. And the symptoms of physiological maturation are, here the cob sheath turns straw yellow color and funicular degeneration takes place and the formation of Duncan layer. This is nothing but a Duncan layer. So this is nothing but a Duncan layer, and the moisture content of the seed is about 35%. This is the symptoms of physiological maturity. The next one is harvesting. After attaining physiological maturity at 30 to 35 days after 50% flowering, and the crop attains physiological maturity 30 to 35 days after 50% flowering. And the seed moisture is at this stage will be 25 to 30%, and the crop is harvested as cob harvesting when the sheath of the cob dries and turns to straw yellow color. Once the crop is harvested, once over harvest for seed purpose. And dehusking, after harvest manually the sheaths are removed is known as dehusking. Manually the sheaths are manually the sheaths of the cobs are removed. This is nothing but a dehusking. And cob sorting, based on the kernel arrangement on the shank as irregular, discolored, diseased and infilling the crops are sorted out, and cobs with the characteristic kernel color and shank color and regular row arrangement are selected for seed purpose. And kernel discoloration should not 10% for certification. There are two different criteria in case of maize as Zenia and metazenia effect. Here the discoloration in crops may be due to disease, infection or genetic contamination. In case of genetic contamination, the effect of foreign pollen on kernel color is known as Zenia. Metazenia effect, which causes genetic contamination in the seed lot. And Zenia is nothing but effect of foreign pollen of same generation, and effect of foreign pollen in next generation is nothing but a metazenia effect. This is how the metazenia and Zenia effect will color. The color changes in the cob. And the next one is shelling. Here the cobs are dried under sun and threshed with a pliable stick for extraction of seeds, and the moisture content at the time of threshing is 15 to 18% only. On large scale production, cobs shellers are used, but care should be taken to avoid mechanical damage, which in turn will reduce seed quality and storability. And the next one is drying. The seeds are dried to 8 to 10% moisture content either by sun or by mechanical dryers for long-term storage, as the seeds are orthodox in nature. Processing. Mechanical grading should be done with cleaner cum grader to remove the undersized, immature and chaffy sheets, and it will vary depending upon the variety. The next one is for seed treatment for storage. Bevestin 2 gram per kg of seed with Cobril 200 milligram per kg of seed as slurry treatment, and bifenthrin at 5 milligram per kg of seed, or this is for better seed storage. Kindly go through this chemicals. And the next one is seed packing. Seeds are packed in gunny bag for short-term storage, and high density polyethylene or poly-lined gunny bag for long-term storage. It can be stored up to 12 months, and for long-term storage it is stored up to 3 years if the seeds are packed in moisture containers and stored at low temperature. The major problem here is incident of grain weevil, which will powder the seed material in short period, and the seed yield is about 3 to 4 tonnes per hectare. And the seed standards in case of In previous slides, we have seen the field standards for seed certification. Here we'll see the seed standards for seed certification. The pure seed percentage is 98% for both foundation and certified seed, and inert matter 2%. Other crop seed 5 kg for 5 per kg of seed for foundation and certified 10 per kg of seed. Weed seed should be none. And other distinguishable varieties 10 per kg, here the 20 per kg, and the germination percentage for varieties in maize is 90%, and the moisture content for seed storage is 12%, and for vapor-proof containers it is dry to a moisture content of 8%. The mid-storage corrections as follows. When germination falls below 5 to 10% of the required standard, seeds are imposed with mid-storage corrections. That is, the seeds are soaked in double the volume of 10 power minus 4 molar solution of potassium dihydrogen phosphate, which is 3.6 milligram per liter of water, and soaked there for about 6 hours, and the seeds are dried back to their original moisture content of 8 to 9%. This is the mid storage correction. And so far we have seen the varietal seed production in maize. In further videos we will see in the next videos we will see the hybrid seed production in maize. Thank you friends. Thank you for watching. Kindly subscribe our YouTube channel to get the upcoming videos.